Bismillahirrahmanirrahim. Assalamu alaikum. Today we'll discuss about the histology of neurons and ganglia. Learning objectives. At the end of presentation, students should be able to classify different types of neurons, differentiate between sensory and autonomic ganglia, and then finally draw a label diagram of ganglion. So before moving on, let's discuss that what is the nervous tissue. The body is composed of nervous tissue, which is also called as nerve tissue or neural tissue. Then it consists of two main important cells, which are the neurons and the supporting cells. The supporting cells are basically to protect, nourish and maintain the nerve cells and their processes. In the peripheral nervous system, the supporting cells are known as Schwann cells, whereas in ganglia, they are known as satellite cells. So, the nervous tissue is also called as nerve tissue or neural tissue, which consists of supporting cells and neurons. The supporting cells are basically to provide nourishment, and then the neurons, which are the structural and functional unit of nervous system. The nervous system has two main functions. One is of excitability, that they respond to physical or chemical stimuli by generating electrical impulses, and then conductivity, that it transmits and receives signals. So neurons are basically the structural and functional unit of nervous system. The main function is to respond to the stimuli by generating electrical impulses. So they are excitable cells, and conductivity as it transmits and receives signals. And the neuron basically composed of three parts: the cell body, which is also known as perikaryon, dendrites and axon. Now we'll move on to discuss brief functions of each part of the neuron. So here you can appreciate these are the dendrites, which receive signals from other cells. This is the cell body, which keeps the cell functional. Then here a big nucleus, which is controlling the entire neuron. Cell membrane for protection. Axon hillock for generation of impulse, and this yellow one is actually the axon, which is running throughout, and this is the axon terminals, which are forming synapses or junctions with other cells. So this is the whole axon, which is covered by the myelin sheet, and the myelin sheet is produced through the Schwann cells. In the center you can here appreciate the nucleus of the Schwann cells, which are the concentric layers and producing the myelin sheet. Then in between we have nodes of Ranvier, which allows diffusion of the ions. So this is basically the structure of neuron with its functions, from dendrites to cell body to nucleus, membrane, and then whole of the axon. Now we divide the neurons into three types. Morphological classification means according to shape, according to function, and according to the length of axon. Morphological classification, also known as classification according to the shape. Here the neurons are basically divided into three types: unipolar, bipolar and multipolar. The unipolar neuron, uni means one, was previously known as pseudo-unipolar neuron. It is called as pseudo because it gives a single process, then divides into a T-shaped manner. So the unipolar is also the pseudo-unipolar with one axon, and it is found in the dorsal root ganglia of spinal nerves and sensory ganglia of the cranial nerves. Bipolar means two. The cell bears a single axon and a single dendrite on the opposite poles, the neurons of which are found in cochlear and vestibular ganglia, retina, and olfactory epithelium. Then the multipolar neurons. The majority of the body is comprised of the multipolar neurons, and they are found most commonly in the pyramidal cells of the cerebral cortex and Purkinje cells of the cerebellar cortex and anterior horns of the spinal cord. So these are the three types. Unipolar was previously known as pseudo-unipolar because of its T-shaped pattern, giving a false appearance. Then the unipolar neurons are found in the dorsal root ganglion, spinal nerves, and sensory ganglia of the cranial nerves. Then the bipolar neurons, bi means two, one axon, and one part is for the dendrites, and they are found in the cochlear, vestibular ganglia, retina, and olfactory epithelium. Then the multipolar neurons, which are found in the pyramidal cells of the cerebral cortex and Purkinje cells of the anterior horn cells of spinal cord. Then the functional classification, that is according to the function, sensory, motor, and the relay neuron. The sensory neuron receives information or stimuli from the environment and pass them to the CNS. Whereas the motor neuron receives information from the CNS and passing it to the effector cells, that could be a muscle or gland or to other neurons. And the interneuron is basically the intermediate station or the relay neuron. Our body comprises mostly 99.9% of the relay neurons. So it gets the information from the sensory neuron, process over here, and then passes on the information towards the motor neuron, completing an arc. You receive an information from the environment, or you get a sensory stimuli which is passed on to the intermediate or relay neuron towards the CNS. Then from the CNS the information is received back that what has to be done towards the effector organ. So the functional classification into three types: sensory, relay, and the motor neuron. Then we have divided it according to the length of axons, Golgi type 1 and Golgi type 2. The Golgi type 1 possess many dendrites and have very long axons. Example, sensory and motor tracts and pyramidal cells of cerebral cortex and anterior horn cells of the spinal cord, whereas the Golgi type 2 neurons have many branched dendrites but have a relatively short axon. So the basic difference is in Golgi type 1 you have a long axon, whereas in Golgi type 2 you have a short axon. And these are basically the interneurons that connect with adjacent neurons. So in Golgi type 1 you have a long axon found in sensory and motor tracts. In Golgi type 2 you have comparatively short axon. Examples are the interneurons. Now we move on to ganglia. So what actually is a ganglia? It is a collection of nerve cell bodies outside the central nervous system. A collection of nerve cell bodies outside the central nervous system. It is basically ovoid in structure. This is the ganglia, and here we have the collection of neuronal cell bodies outside the central nervous system, supported by connective tissue. So it is an ovoid shape, oval structure containing neuronal cell bodies outside the central nervous system, known as ganglia. The function is that it serves as a relay station and transmit impulses. The nerves giving information and taking them out. So microscopically they are divided into two broad types, sensory ganglia and the autonomic ganglia. Before going on to each with details, the both type of ganglia have some features in common, like a connective tissue capsule which is covering the whole of the ganglia. This is the connective tissue capsule, and it is continuous with the connective tissue throughout the substance of ganglion. The body of each ganglia is enveloped by small flattened cells known as satellite cells, which I have already mentioned in the very first slides, that the nervous tissue is divided into two main cell types, the neurons and the satellite supporting cells. The supporting cells in the ganglion are known as satellite cells, which basically function as a metabolic exchange of ions and for nutrition and protection as well. So these features are in common, like every ganglion will have a neuronal cell body and a capsule, and every ganglia will be covered by the satellite cells. So the ganglia are the ovoid structures containing the neuronal cell bodies, divided into two types, sensory ganglia and the autonomic ganglia. Now we'll move forward to each of the type. First of all, sensory ganglia. The sensory ganglia receives the impulses from periphery and convey them towards the CNS. Like what was sensory neuron doing? Receiving information from the environment and giving it to the CNS. Similarly, the sensory ganglia will receive impulses from the periphery or the environment and will convey them towards the CNS. The sensory ganglia, also sometimes known as the peripheral ganglia, So the sensory ganglia receive information from the periphery and going back towards the CNS. Under the microscope, what you can appreciate and what is important for you is, this is the ganglionic cell body. Then we have the flattened satellite cells, these black one you can appreciate is covering the whole of the cell body, ganglionic body. The neurons are of pseudo-unipolar type and the nucleus is central. The nerve fibers passes through the ganglion. So the nerve fibers are passing here and there. Here you can also appreciate a single cell or a ganglionic cell, which is covered by the satellite cells. The nucleus is central and the neuron is of pseudo-unipolar type. Whereas in autonomic ganglia, you can appreciate that this is a ganglion. It has fewer satellite cells, whereas in sensory ganglia it was totally covered. It has fewer satellite cells. It has multipolar neurons. So sometimes it looks like a star shape. The neurons are scattered. As in the previous, it was closely packed. Here the neurons are scattered. And the nuclei is eccentrically located, whereas in sensory ganglia it was central, but in the peripheral or the autonomic ganglia it is eccentric, means on the side. The autonomic ganglia is further divided into sympathetic and parasympathetic divisions. The parasympathetic is somehow found in the walls of the viscera and may be devoid of a proper corrective tissue capsule. Now here you can again appreciate sensory and autonomic ganglia. This is what will be what you can see under the microscope. This kind of picture would be appreciated. Then when we have magnified it, this is the central. The bodies are closely opposed, all of the bodies, there are few gaps in between. Then the nucleus is central. Here also, here also, here also, you can see the nucleus is central, plus it is a pseudo-unipolar neuron, a unipolar or a pseudo-unipolar neuron, then a complete ring of the satellite cells, plus there are no synapses. Whereas in autonomic ganglia, you can already appreciate it's some kind of a star-shaped multipolar neuron, where the cell bodies are not closely packed, they are scattered, here, 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 they are scattered, and there is an incomplete ring of satellite cells. Very few satellite cells can be appreciated, and synapses do occur. So the differences are very important for you to learn. Now here again the differences between the sensory and motor ganglia. Sensory was large, rounded, pseudo-unipolar. Motor was small with multipolar neurons. Nucleus centrally placed. Nucleus eccentrically or peripherally placed. They are found in groups, they are found scattered. They have a well-defined satellite cell and poorly defined satellite cells. The examples are the spinal ganglion or dorsal root ganglion, and here the autonomic is also known as sympathetic ganglia, whereas the parasympathetic ganglia are found between the muscles of intestine, which you will study inshallah in second year. So this type of diagram can be made on your practical copies, where you can see these are the unipolar or pseudo-unipolar neurons, each of which encircled with the satellite cells, and the nucleus is centrally placed, and the nerve fibers are passing through between them. This is here, it is the structure of the satellite cell, how it looks. And whereas in sympathetic ganglion, the multipolar neuron, star shape, the nerve fibers going here and there, this is actually the capsule. The nucleus is peripherally placed, and few satellite cells. Do make satellite cells as well, which are few in number and not completely covering. Okay, so that this might help you. Now for the assignment, you have 2 simple MCQs and then an SEQ, classification of neuron, 4 differences between sensory and autonomic ganglia. It's a very easy assignment. Then finally you have to draw and label histological section of both sensory as well as autonomic ganglia. So this was whole about the lecture. Hope you have understood everything, and for queries you can always ask me. Thank you.